Hi, my name is Shelby Boone, and I did my entomology project on the effects of isopentyl acetate on honeybee defensive stinging. Firstly, we're going to take a look at honeybees, also known as Apis mellifera. Apis mellifera is the scientific name for the western honeybee. It's Latin for the term honey-bearing. So honeybees are very social insects. They rely on communication for many different reasons, whether it be mating, foraging, defense, daily operations, jobs, and more. Without communication, a hive would be a chaotic and disorganized mess. It helps keep things stable and things to run smoothly. But how do bees communicate? Bees use pheromones to communicate. These are chemical signals that are let off for whatever reason a communication needs to occur. One type of communication is an alarm pheromone. When a hive is threatened or a honeybee senses a danger, an alarm pheromone is given off, which alerts the colony that danger is present and that action needs to be taken immediately. We're going to take a close look at an alarm pheromone known as isopentyl acetate. Isopentyl acetate, also known as IPA, is the primary alarm pheromone in honeybees. It is released when a honeybee feels threatened, and it signals other bees to come help defend and attack the threat. IPA is produced in the mandibular gland and is released near the stinger. Honeybees will use their wings to spread the pheromone nearby to alert their hive mates of a threat. The image here on the right is a view of the mandibular gland from the side. You can see it highlighted in red. The bottom image is the chemical structure of isopentyl acetate. I came up with the research question, how does isopentyl acetate affect a honeybee's defensive behaviors? The first study I found looks at the effects of different amounts of isopentyl acetate, or IPA, on honeybees. Researchers did 5 different experiments, each with a different composition of IPA with paraffin oil. These experiments range from low IPA concentration to high concentration, the lowest being a dilution of 1 in 10,000, and the highest was 1 in 2. Researchers used paraffin oil because it causes no additional response and reaction in honeybees. They measured reaction time, duration of response, and the strength of response. The main finding is that higher amounts of IPA showed increased responses in honeybees for longer amounts of time with stronger defensive reactions. The table on the left shows each experiment done in order left to right, going from lowest concentration of IPA to highest concentration of IPA. When it came to seconds to react for a honeybee, the lower concentration showed a time of about 41 seconds to react to a defense, whereas the higher concentration of IPA showed 8 seconds, which is a significantly shorter time. We also see the duration of reaction vary greatly. The lower concentration showed 10-second duration of reaction, whereas the higher concentration showed almost a minute of a reaction time at 55 seconds. This proves how IPA causes a faster reaction time for a longer amount of time. The second study I found shows isopentyl acetate being compared to another pheromone for alarms called 2-heptanone. This is also known as HPT, and it's known to be a less effective alarm pheromone when compared to IPA. Researchers were curious just how much better IPA is compared to 2-heptanone. Honeybees were exposed to different amounts of each chemical, and then their defensive behaviors were observed and recorded. Once again, they measured the intensity of their response, the amount of response, and the effectiveness of different amounts of each pheromone. The main finding in this study was that IPA showed to be far more effective than HPT was at increasing defensive behaviors in honeybees. The graph here on the right shows the concentration of IPA on the Y vertical axis and HPT on the horizontal x-axis. It shows a far higher amount of IPA than when compared to HPT, which proves how honeybees were far more responsive to the IPA than the HPT. The third and final study looked at the behavioral effects of IPA on brain activity and gene expression. Researchers exposed IPA to honeybees once again. They observed the immediate effects on behavior, but they also recorded their behavior during later exposures. They looked closely at changes in gene expression in honeybees' brains. They specifically looked at the expression of the gene c-Jun. c-Jun is in charge of how bee brain responds to stressful signals. The main finding was that IPA showed to give honeybees an immediate defensive response, but also later affected their brain gene activity and behavior. So these graphs on the left show that IPA does increase defensive behavior and it increases c-Jun expression. IPA lasted into later exposures, showing that it has a long effect on a bee's defensive reaction. With more c-Jun being expressed, it indicates that bee brains are more active under defensive conditions. So IPA is proved to provide long-lasting effects for a bee. In conclusion, isopentyl acetate is a fast-acting and long-lasting alarm pheromone for Apis mellifera. It directly influences defensive behaviors in honeybees. It can cause faster reactions, higher aggression levels, and more readiness and alertness. It is the primary alarm pheromone and helps future reactions in honeybees through c-Jun activation. There is much significance in our findings. It shows how honeybee colonies can defend their hive, shows us how the main chemical, isopentyl acetate, causes strong defensive behaviors. It proves how behaviors are connected to activity in the brain and genes. It shows us how honeybees can survive threats and predators. And it shows how this is all part of the ecological system, and how honeybees, while they're very small, they can be quite mighty with their defensive behaviors. These are my references. Thank you so much.